afternoon, everyone, and welcome to today's PHO Microbiology Rounds presentation on building a pediatric infectious disease observatory in a resource-limited setting. My name is Vanessa Tran, and I am a clinical microbiologist at Public Health Ontario, and I have the absolute pleasure of moderating today's session. Before we begin, we would like to acknowledge that the land that we are gathered on is the traditional territory of many nations, including the Mississaugas of the Credit River, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples, and is now home to many diverse First Nations, Inuit, and Métis peoples. Toronto is home to Indigenous peoples from across Turtle Island, and we are thankful for their hospitality that allow us to live and work today. It is now my pleasure to introduce our speaker for today's event, Dr. Senjuti Saha. Dr. Saha serves as the Deputy Executive Director of the Child Health Research Foundation in Bangladesh, leading research on pediatric diseases. She earned her PhD in molecular genetics at the University of Toronto, followed by postdoctoral training at the Hospital for Sick Children and Stanford University. Since 2016, Dr. Saha has pioneered genomic research in Bangladesh. She contributes to several influential global health committees, including the WHO Product Development for Vaccine Advisory Committee, the Technical Advisory Group on Bioevolution, and the Polio Transition Independent Monitoring Board. Out of breath. With over 80 peer-reviewed publications, Dr. Saha also serves on the editorial board of several international journals. In addition to her research leadership, Dr. Saha leads a nationwide program called Building Scientists for Bangladesh, advocating for equitable science, education, and opportunities. And with that, I will pass it over to Dr. Saha. Thank you very much.
Thank you, Vanessa, for that very, very generous introduction. It is an absolute pleasure to be here today and be able to share some of the work that we have been doing in Bangladesh. It's a privilege that I am here representing the work, but I will show a picture of some of our team members. So really the work that I'll be showing today is the contribution of many, many people over decades. So I'm going to start by showing this PHO disclaimer slide. I'm going to move on to say that I work at the Child Health Research Foundation. I sit in multiple committees, and the Child Health Research Foundation is a nonprofit organization who receives grants from different country governments and other for-profit, nonprofit organizations. I hope that today, by the end of the session, the participants will be able to identify challenges that we face in building infectious disease research capacity in settings like Bangladesh, where we have access to very limited resources. Participants will be able to explain the use of genomic tools and how the data that comes out of using genomic technology can actually be used for evidence-based policy decisions, specifically microbiology related policy decisions. We will discuss the impact of local genomic research and again how that can help aid public health responses. And finally, participants will be able to recognize the value of capacity building. With that, I'm going to go on to my first slide. So I work in a very small country called Bangladesh. It's right over here, shown in red. And this is how it compares to Canada. So much, much smaller. However, when it comes to people, we are not that much smaller. So this is how Bangladesh compares to Canada. Canada is almost 10 million square kilometer with a population of about 41 million. The density, population density in Canada is about 4 people per square kilometer. Now, how does that compare to Bangladesh? We are about 148,000 square kilometers, so much, much smaller. However, we have a population of over 170 million people, and our density averaged out over the entire country is about 1,100, closer to 1,200 per square kilometer. I spend most of my time in the capital city of Bangladesh called Dhaka. It's one of the most densely populated cities in the world with about 34,000 per square kilometer. So we know each other, and you'll be surprised to know how many people we get to know and become friends with. Perhaps despite this or in spite of, we have done really, really well when it comes to public health and implementation in Bangladesh. We are a very pro-vaccine country with vaccine coverage over 90% across the country, even in very hard to reach areas. So mothers are very careful about ensuring that their children are vaccinated. We have amazing community health workers, mostly led by women. And we really have achieved all 3 health-related Millennium Development Goals way before schedule, and our government received multiple awards for that. I and my team, we have the privilege of working in this country. And as I said, our organization is called the Child Health Research Foundation. We are a nonprofit working in the country for over 30 years now. And our mission is to improve child health by facilitating appropriate policy decisions through research and advocacy, which means we work in the lab, we work in the hospitals, but we also have a large advocacy team who takes the findings of labs and hospitals and brings it to the policymakers such that they understand and they're able to make decisions based on the information produced. And we work with both national and international policymakers. So a little more of a history. The first lab that CHRF started was in 1989 when Professor Shomir actually started the first microbiology lab in the largest pediatric hospital in the country. A lot of the data that I will be showing you today come from this large hospital in the country called Bangladesh Shishu Hospital and Institute. It is a tertiary level hospital, the largest tertiary level hospital for children, where we see children from all across the country. So while I said that, our headquarters is in Dhaka, which is right in that big location marker in the middle of Bangladesh. So that's the capital city. This is exactly the building where my main office is, and all our research labs are there too, molecular microbiology, etc. However, in addition to our research facility in this building, we coordinate our work across 4 different laboratories, and these laboratories are located in 4 different hospitals, as you can see, in different parts of the country. That includes both rural hospitals and urban hospitals. In addition to working in hospitals, we know that majority of the children in resource-limited settings actually do not show up at the hospital when they fall sick. So in order to understand true burden of diseases, we also do house-to-house surveillance, where we visit homes of children under the age of 5 years every week and try to understand the burden of pneumonia, meningitis, and sepsis. I'll talk about this more later, but this is called a demographic surveillance system. We, of course, do not do it across the entire country, but in a small population of 400,000 in a rural region surrounding one of our rural hospitals shown over here. So really leveraging this platform of network, surveillance network, diagnostic network, and research network, we specifically focus on systemic infections. We focus on neurological infections, so that's primarily my interest. I moved to Bangladesh about 10 years ago, and I specifically moved there to look at idiopathic neurological infections or idiopathic meningitis. But that is not something we'll be talking about today. We'll be talking about systemic infections. In addition to systemic infection, neurological infections, the main cause of mortality due to infectious diseases is respiratory infections. So pneumonia is still the largest killer in many countries, including Bangladesh. We conduct surveillance research on these 3 kind of broad areas, and the data generated are translated to policymakers. So it was actually data generated from this platform that convinced the Bangladesh government and Gavi and international policymakers to introduce the Haemophilus influenzae type B, or the HIB vaccine, in 2009, and the pneumococcal vaccine, we have PCV10, in 2015. As I was saying, today we'll be primarily focusing on systemic infections. So in 2004, by when most of our network was already set up, we set out to conduct surveillance on systemic infections in children. And this surveillance kind of had three broad objectives. The first objective is to describe epidemiology and estimate burden, both in hospitals and community. As I've already mentioned, to characterize what are the bacteria that are circulating and causing infections. So, genotypically and phenotypically characterizing them, understanding that their antimicrobial susceptibility patterns, etc. And then kind of using this platform such that we generate baseline data, but if interventions like vaccines are introduced, we are also able to study the impact of those interventions. So this platform was leveraged to understand the impact of the Hib vaccine, which is, as we all know, a miracle, wonderful vaccine. And currently we're looking at the impact of the pneumococcal vaccine in pneumococcal diseases. Now, this is a slide, it's a busy slide, but I will pick some important things from here. But what I am trying to show you in this stacked bar graph is basically the bacteria, specifically focusing on gram-negative bacteria that we have been detecting over the last 18 years or so. So you can see that in different colors are different bacteria, and with time, there has been an increase in the number of bacterial isolates we are seeing in our hospitals. In red, you'll specifically see Klebsiella. So Klebsiella infections since 2015, 2016 have really been increasing. And this has been very striking for our team. Why are Klebsiella infections increasing? What is the mortality associated with Klebsiella infections? Who is the main victims of this bacteria? So we started really analyzing data from the last 18 years, and that is what I am going to present today. I'm going to talk about the rising problem of antimicrobial resistant infections, Klebsiella infections, in neonates of Bangladesh. So in neonates are babies in their first 28 days of life. So just a quick review of Klebsiella species. So Klebsiella is a very common bacteria. It's ubiquitous, found everywhere, this bacteria, and it is mainly known to cause pneumonia, sepsis, wound infections, UTI in adults. But it specifically kind of affects health of two extremes, either very young babies, the elderlies, and also immunocompromised patients, so patients in hospitals. So very common hospital-acquired infection, Klebsiella also is. It encodes various virulence factors that helps it really cause severe infections. And it's also known to be antimicrobial resistant. So this is what the outer membrane of Klebsiella looks like, and I'm going to come back to this towards the end of my talk. So there is an outer membrane, which basically has the O antigen, the LPS, and the capsular antigen, also known as the K antigen, and we will come back to this in a bit. So for the longest time, as we studied microbiology in like older books of Klebsiella, we thought Klebsiella pneumoniae was the main species, and we are able to detect it in the laboratory through traditional tube biochemical tests. But with the expansion of genomics of bacterial whole genomes, we now know that Klebsiella pneumoniae is only one species of a Klebsiella pneumoniae species complex. In addition to Klebsiella pneumoniae, there are 6 other species that form part of the same complex, and Klebsiella pneumoniae is only one of them. It's not to this date, it's not possible to distinguish in the laboratory Klebsiella pneumoniae from all the other species in the complex in using traditional biochemical testing. In order to differentiate between these bacteria, we have to do whole genome sequencing. Now, in Klebsiella pneumoniae, there are hundreds of clones that are circulating across the world. Some cause infection, some don't. A proportion of these clones contain multi-drug resistant genes that lead to, of course, antimicrobial phenotypic antimicrobial resistance. And then some of these MDR strains become local problem clones. So, in Bangladesh, there are certain clones that are spreading across the country, and I will show you data for that. In China, these are different clones. But in many cases, local problem clones, which are not always necessarily antimicrobial resistant, basically overlap with also clones that contain a lot of multi-drug resistant genes, and they spread across the world. And those are called global problem clones. So these are highly virulent strains that have very good fitness, are able to spread locally, but also acquire multi-drug resistant genes, and then they become global problem clones, leading to high mortality. As I said, or as I showed already in one of my first slides, that the burden is increasing. How do we know that? Since 2004, in our surveillance that I was talking about, we have been enrolling every child who walks into our network hospital, and we are trying to see if they meet the case definition of pneumonia, severe pneumonia, sepsis, or meningitis. We collect all of their information, right? Demographic information, age, sex, clinical information, how long did they stay in the hospital, what was their hospital outcome. In some cases, we also do home follow-up. Of all the 122,000 cases, we did blood cultures using BACTEC in recent years. And what we found is that in these years, there was 1,600 positive cases of Klebsiella. We did antimicrobial susceptibility testing on all of these isolates. And then I will go on to genomics in a bit. We selected a subset of isolates that cause infections in young babies and looked at their genomic or genotypes and genomic characteristics. So starting with age, as I was saying, Klebsiella really has this propensity of infecting very, very young babies whose immune system is not completely developed yet, and in many cases, perhaps preterm babies. So this shows basically that babies in their first month of life, or neonates, are most susceptible to infections by Klebsiella. And over here, I've broken up the first 2 months of life into days, and you can see it's really the first 10 days of life when babies are most susceptible to Klebsiella infections. The burden of Klebsiella infection is increasing. Now, initially, I just showed you a bar chart, but there are many caveats to that, right? Our hospital capacity is increasing from 400 beds. 30 years ago, we have now 665 beds in one of the largest hospitals. So really, we have to have a denominator. So what I'm showing you over here are the number of Klebsiella cases per 1,000 babies or children, actually, that were enrolled in our surveillance network. And it is very clear that the burden of Klebsiella infections is increasing, and we kind of see this upward trend primarily starting in 2014, 2015. When we look at mortality caused by Klebsiella infections, we see that not only since 2014, 2015, not only the number of infections is increasing, we also see much higher mortality. So before 2015, we saw deaths odds ratio of 1 of mortality, but we see that in the last few years, mortality have increased by over 4 times. So in 2021, mortality was 4 times higher than, let's say, in 2004. So we see this high rise of mortality also. And very interestingly, this increase in mortality actually overlaps with increase in carbapenem resistance of Klebsiella pneumoniae, which is one of the last line of drugs to treat these infections. So what we see over here is that around 2014, 2015, most of the isolates we were finding in these babies were resistant to carbapenem. So currently over almost 80% of all isolates of Klebsiella are resistant to carbapenems. And it's around the same time that we started seeing this increase in mortality also. Now, of course, this is not showing causality, it's just showing association. So then we started doing some analysis to look at this association more closely. And what I'm showing you here is case fatality rate by Klebsiella infections in the pre-2015 era, shown over here. And we see that there was really not much of a difference between mortality due to carbapenem resistant strains and carbapenem sensitive strains, shown over here. So this is case fatality rate. In both cases, it was between 10 and 15%, let's say. So there was no significant difference between mortality caused by the two different groups. However, starting in 2015, we see a marked increase in mortality due to carbapenem-resistant strains. Having said that, though, we also see increase in mortality due to carbapenem-sensitive strains. However, now the difference is significant. So carbapenem-resistant strains is more likely to kill a baby than a carbapenem-sensitive strains. But what this tells us is that resistance is not the only factor that is leading to higher mortality. There might be other things that this bacteria is acquiring or becoming more adapted to cause virulent infections, highly pathogenic infections that has happened in the last 5-6 years or so. So there's definitely things that are changing within this bacteria. And that is really where genomics come in. So as I said, I moved to Bangladesh about 10 years ago. And one of the primary objective that I had was understanding neurological infections, idiopathic neurological infections, for which we set out to establish a state-of-the-art genomics facility. So that genomics facility was set up in 2018, and leveraging that, we started sequencing Klebsiella from the last 20 years and studying them over time. And this was possible because our founder, we also call him, well, I call him a bacteria hoarder. People tell me hoarder is not the nice word, so now we call him a bacteria curator, where since he established the lab in the hospitals, he started basically keeping every bacteria that he found. And with time, this entire biobank became digitized, where every bacteria basically goes into this glycerol stock. And I don't know if you can see it, but every tube is barcoded. And you scan that barcode, it immediately gives you all the information about the patient that bacterial isolate came from, age, demographic, outcome, all the information that we collect in our surveillance. And we have a lot of -80s filled with over 100,000 samples, actually, as I speak, either, samples from the patients or pathogens that were isolated from these patients. So this has been this beautifully biobank giving us this very rare opportunity of looking at historical trends of infections and different evolution of bacteria. So this is what a part of a genomics facility looks like. It's bigger now. We have 3 sequencing machine. This is the machine. I wanted to show you a picture of this machine because all the data that we generated, in addition to the people, were actually generated by this machine. All the work was done in this room. We also call this room the CHRF Infection Observatory, because this is the room where samples are prepped, viral, bacterial, or just raw samples of, let's say, cerebrospinal fluid samples, and they go into this machine that generates the data that we are able to interpret later. So for specifically Klebsiella infections, the genomic analysis was led by a senior scientist called Dr. Yogesh Huda. And the data that I'll be presenting now is really because of the amazing work that he has done over the last couple of years. And this work has been funded and continues to be funded by the Bill and Melinda Gates Foundation. Actually, it's just the Gates Foundation now. So I told you there were like 1,600 cases that we isolated or detected of Klebsiella infections in the last 18 years. So we kind of took a representative number of Klebsiella from all the previous 18 years and sequenced them. So in dark blue are the samples that we sequenced, in light blue are the samples that we did not sequence. And overall, it comes to like 40% of all the strains that we sequenced. You see that we couldn't sequence too many of 2004 and 2005 because some of the bacteria we could not revive anymore. So after sequencing, we immediately analyze them to understand what are the most common species of Klebsiella that we are finding in our population. No surprise at all, the most common species is Klebsiella pneumoniae, but 15% of the isolates are actually made up by Quasi-pneumonia subspecies Simile-pneumoniae or Quasi-pneumonia subspecies Quasi-pneumoniae, Varicola, and also Aerogenes. So really, it's a mix. For the longest time, we thought it would be all or majority Klebsiella pneumoniae, which is true. Majority is Klebsiella pneumoniae, but when you have such a high burden of infection, 15% by other species is also a very large number and definitely demands attention. We looked at, are there like significant variations through time of how this species change or the population, composition of the different species, but we do see that there isn't really any significant change. Majority of the species every year is Klebsiella pneumoniae. And then we looked at mortality by different species of bacteria, and we see that Klebsiella pneumoniae really causes the highest mortality, as I showed you before, almost close to 50%, but others do also cause high mortality, right? So there are other groups now at our foundation who are focusing on different species of Klebsiella species complex, pneumonia species complex, but for the rest of my talk, I'll just focus on Klebsiella pneumoniae. So we sequenced them, and like every other group, we made a beautiful phylogenetic tree. So what I'm showing you in this phylogenetic tree is that there are two circles, circle number 1 and circle number 2. And circle number 1 are the sequence types that we detected in all our Klebsiella pneumoniae, and circle number 2 is basically years when they were isolated. There are a couple of things that became obvious very, very immediately, and it's that these are not just outbreaks. It's not like one clone is causing all the infection in Klebsiella pneumoniae, because there is this idea that all Klebsiella pneumoniae infections are hospital acquired, and it all depends on what clone is circulating in your hospital at that time or over time. Looking at this phylogenetic tree, it immediately became clear that it's not one single outbreak that we can immediately tackle. Now, phylogenetic trees are very, very beautiful, and I'm showing this to you to convince you that we are, or my team is able to make phylogenetic trees, but I'm just going to go back to a bar chart, which is a lot more informative, and I'm going to kind of like show us this dance of different sequence types over time in Bangladesh. So if I orient you a little bit, over here is again, time on the x-axis and proportion of different sequence types of Klebsiella pneumoniae we're seeing in these babies. And every color in these bars is a different sequence type shown over here. So, and then on top is basically the number of isolates we sequenced per year. So again, this makes it even perhaps more obvious than that phylogenetic tree that indeed it's not one sequence type that's causing all the infections. It's not one sequence type that's just taking over throughout the entire year. It's not one sequence type that we can tackle or we can clean up the hospital and stop all infections. There seems to be circulation of different sequence types. And what I'm not showing you over here is that these are bacteria coming from different places of the country and at least 4 different hospitals. And these are also babies that have tested positive for Klebsiella pneumoniae within 48 to 72 hours, mostly of hospital admission. So also most likely not hospital-acquired infections. What is also very interesting is that the sequence types rapidly change every year, and that's why I call it the dawns of sequence types. So over here is an example of 2020, and you see there's this specific sequence type called ST11, a very well-known global problem clone. Documented by the WHO that this is indeed a global problem clone, ST11. And we see that in 2020, even in 2019, this was indeed the most abundant or the predominant sequence type circulating. However, the moment we move to 2021, the colors are not coming up very well, but we see that ST11 has almost disappeared. Now we have data from 2022 and 2023 and 2024, and ST11 has completely gone, replaced by a variety of other sequence types. So there is this constant dance of different sequence types, and we really don't understand how, why, when one clone on one sequence type takes over the others. Now, if I show you the sequence types across the world, you will also see a lot of diversity. So for this map, what we did is basically we took the top 10 most common sequence types of Bangladesh in the last 18 years, and we kind of tried to look at their distribution across the world. Are there many similarities? Are they completely different? And it is very clear that our top 10 sequence types makes up a different size of the pie in different parts of the world. So, maybe a quarter in Europe, a quarter in North America, but a lot more than that in East Asia, right? And even in India, which is a neighboring country, it only makes up about a quarter. And we also see ST147 primarily there instead of ST11, which was a predominant clone. Now, of course, there are caveats in the figure that I'm showing you over here. Caveats include there has been no systematic longitudinal study to look at changes in sequence types. I am showing you data from 18 years. Most of the data from other countries other than Europe and North America are sporadic. They come from, 1 year, some come from adults, some come from 1 or 2 hospitals, some are countrywide, some are just one city. So there are definitely a lot of gaps in this data. But what we can take away from here is that there's a huge diversity of Klebsiella pneumoniae circulating across the globe. And this is something we have to keep in mind as we move forward thinking about interventions, and we can discuss this during the discussion session. What we also see is that carbapenem resistance that we see in all of these strains, or at least the newer strains of Klebsiella pneumoniae, is not like one gene that's causing all the resistance. Carbapenem resistance clearly was acquired, or it emerged many, many times in different clones. We see NDM1, we see NDM5, NDM7. So again, there are some common genetic markers of carbapenem resistance, and there are some sequence types that are more likely to carry certain resistance markers. But again, there's a lot of diversity that we see in what causes these strains to become carbapenem resistant. Again, adding to the evidence that it is just not one clone that's circulating in the country causing all the infections. Now, I'm approaching the end of my talk, and I wanted to perhaps talk a couple minutes about how can we prevent these infections. We can discuss different types of interventions, but because I showed you genomic information, I must tell you that different interventions are being designed that's pharmaceutical, and most of them are kind of targeting the capsule of Klebsiella pneumoniae. So over here, I'm going to bring you back to one of the first slides that I showed, that this is Klebsiella. It has the K antigen and it has the O antigen. So vaccines or monoclonal antibodies are currently being designed against the K antigen, which kind of hides the O antigen. So antibodies targeting O antigen don't work very well. They're kind of hidden. So it looks like K antigen it will be a better target for developing monoclonals or vaccines. So what does the diversity of O antigen or Ko antigen look like? So O antigen, which is, LPS kind of covered by the K antigen, we see that if we took the top 10, we would cover 92% of all the O antigens that we saw in Bangladesh over the last 18 years. So diverse, but not so diverse. If we had, let's say, a 10-valent vaccine, something like a pneumococcal vaccine, would probably be able to protect babies from infections. However, as I said, O antigen isn't the best target for such interventions. A lot of work is being done on the K antigen. K antigen, on the other hand, is a lot more diverse than O antigen, and the top 10 only make up 60% of the isolates we're finding. So we'll have a lot less coverage, let's say if there was a 10-valent intervention. And not only that, what is really, really I guess as a molecular biologist, interesting, but also challenging for scientists and companies developing interventions, is that these K antigens are not stable. They change with time. So very similar idea to sequence sites, right? So there are, again, a very similar graph. We have time on the x-axis and then proportion on the y-axis. And this time, instead of sequence types, I'm showing you K antigen types. And there are certain sequence types associated with certain K antigens, but it's not a fixed relationship. And we see that these K antigens are also really changing over time. So which K antigen do we target and when? And do we need to upgrade our K vaccines or interventions constantly? Are questions that are really being discussed by policymakers and donors and the WHO at the moment. So I hope what I have been able to show in the last half an hour or so is that Klebsiella infections are increasing in number and they're increasing in severity. So not only are we seeing a higher burden, we're seeing more babies in their first month of life succumb to this infection. We're seeing increased carbapenem resistance, and this correlates with increased mortality. However, antimicrobial resistance doesn't explain all the mortality. There must be other factors, and we can discuss the factors later, but this is definitely an area that must be investigated of why are sensitive strains also leading to severe infection and mortality. We also saw Klebsiella diversity is very, very high in Bangladesh, and there have been rapid changes observed in the last few years. So we need to really keep track of how types are changing and constantly discuss is really interventions like other vaccines the only way or the right way to, because this has real implications on vaccine design or monoclonal antibody design. I showed you data today on Klebsiella. So this is, again, one of the first slides I showed. But if you look at this graph carefully, it's not only Klebsiella that's leading to more and more antimicrobial resistant infections. We are seeing higher and higher burden of Acinetobacter. So we have now a dedicated group just focused on studying Acinetobacter. What are the species that's causing most common infections, their antimicrobial susceptibility patterns. We are working with a group in Hungary to understand a phage interaction with Acinetobacter, and is phage therapy also an avenue we could potentially look into. So there's a group working on that. And then another surprise has been this increase in Burkholderia. You will see that we barely ever saw Burkholderia in the previous years, but since 2019, we're also seeing an increase in Burkholderia infection. So we are sequencing almost all Burkholderia, answering very similar questions of what's leading to more infections and how can we tackle perhaps these infections better. I'm going to end here, and I'm going to say again that it's a privilege to be able to share this data with all of you, but I do the minimum amount of work, I think, in our organization. We have a wonderful group of 300 people working across different sites, a lot of them in this picture on a rooftop of our research headquarters, and it's an amazing group of people working day and night really to prevent infections and save lives. And our funding for the studies that I showed you today come from the Gates Foundation, Chan Zuckerberg BioHub, and the World Health Organization, who supported our surveillance network for many, many years. I'm going to end there. And if you have questions, happy to take them.
Thank you so much, Senjuti, for that very enlightening presentation. The work is amazing. Don't undersell yourself. I'm sure you're doing an incredible amount of work to facilitate it all. We're now going to open the floor for our Q&A session. So this is your chance to ask questions and hear directly from our expert speaker. If you haven't done so already, please go ahead and submit your questions into the Q&A pod. And with that, we'll begin our Q&A and invite your questions for Dr. Saha. So I have a bunch of questions, but I'm going to start maybe with the epidemiology because there's something that really stood out to me. And it was 2015. It seems like an inflection year where you saw not only like a steep, you began to see like a steep increase, but also when you looked at your genomics, that year was unique in that there were two predominant ST types, whereas there were, I guess, more diverse than the other years. And it was just that 1 year. So it looked like there was something that happened. And I was wondering if you could provide any insights in terms of other factors, travel, socioeconomic, that could have contributed to this market rise after that year.
Thank you. Thank you so much for that question, Vanessa. So absolutely. So 2015 and 2014 was also interesting. Around that time, we kind of also saw a dip in infections. And then we see this rise. So, when we started analyzing that data, we continued to scratch our head, like what happened those years? So we went back to all our data forms, hospital, were there renovations taking place in the hospital that changed the bacterial population circulating? And we haven't been able to find an answer for that. So, very good observation, but I don't have an answer for you at all. And we're trying to really address that in the manuscript that we just submitted too. Like what happened? We don't have an explanation.
Do you know if there is specific, with your phylogenetic analysis, we can look at potential specific importations? I don't know if there was increased travel after that time.
Yeah, so with our data set, we cannot do that analysis. Because a lot of the work that we do is retrospective. So the data have already been collected, and we did not collect any data on travel. But very good question. And what really makes all of this very complicated is we don't still understand the mode of transmission of Klebsiella pneumoniae, right? What we are seeing is that these babies are coming to the hospitals already with, blood full, their body full of Klebsiella, because we're doing blood culture within 24, 48 hours of infection, and we see Klebsiella in blood. And these babies are 2 days old, 1 day old, 3 days old. So is it, are they getting infected right after birth, during birth, in the birthing facility? A lot of births happen at home. We're also seeing an increase in cesarean sections across the country. So we also don't understand that mode of transmission, because if we at least had good idea about that, maybe we could then start answering some of these questions. Have the moms traveled, or do certain areas have more burden of certain sequence types? Yeah, a lot of unknowns.
That's a very good segue to my next question, and you may not be able to answer this, but yeah, around I guess the mode of transition, because these like the infection occurs so quick, so soon after birth, like presumably it's household or mom. Do you have any idea about the prevalence, like the background prevalence of Klebsiella and the ST type, specifically around like carriage in the moms or carriage in the household dwellings?
Again, very good question. We do not have that. We have no idea about carriage in moms, carriage in households. No studies have been done looking at that, but we are really hoping that the data we are showing here will lead to some of those research studies to fill those study gaps. However, there have been a couple of studies done in other countries. There was a very famous a study done in a small area in Italy where they swabbed everything and sequenced every Klebsiella pneumoniae they found. So they swabbed humans, they looked at infections, isolates from infections, specifically in elderlies in that case in Italy. Then they looked at like horse poop, chicken poop, farms, everything, because Klebsiella is everywhere. And what they showed, that study showed, and it's been used as a landmark study, kind of to show that there is a huge distance between what are the Klebsiella that's circulating in the environment and what is the Klebsiella that's causing infections. Now, that's the largest study we have seen, and we cite that a lot, saying that there is a huge difference between pathogenic Klebsiella and non-pathogenic Klebsiella. But we are gradually also getting some evidence, obtaining some evidence that that might not be true in very high burden settings. I mean, we have 34,000 people per square kilometer, very different than, Pavia or, the rural region of Italy where that study was done. So how bacteria circulates is also very different, and region specific, geography specific, also different in an urban area of Bangladesh or rural area of Bangladesh. So a lot to again also study there. The only thing we have done, like one of our master students basically collected sewage water from across Dhaka city to look at Klebsiella, and it was very interesting. She did not find a single carbapenem resistant Klebsiella in sewage water. We do not know why. We do not know, is it because of the way we were selecting, or it was just outcompeting carbapenem resistant Klebsiella, but we did not find it in sewage. So where are they circulating? We don't know.
I'm going to give some of our audience a chance to ask a question.
Yeah.
So there's I guess I'll combine these two questions. So what is the burden of hospital, like nosocomial spread of Klebsiella pneumoniae? And for the ones that are resistant to carbapenem, so meropenem, what is the drug of choice for treatment?
Yeah. So first of all, about the first question was how many of them what proportion of them are nosocomial? So about 70% of all the Klebsiella isolates that we detected were detected within 72 hours of hospitalization. So most likely they're coming to the hospital with the infection. So I can say that the 70% of the infections were not acquired in the hospital where we do the study. That in no way means they were not acquired in a different hospital. Unfortunately, that is data we do not have. The baby could have been born at home or a different maternity unit and then maybe acquired the infection there and came to the hospital. But we see a lot of infections in the first or second day of life. And now I regret not having that slide. But a huge proportion is not hospital acquired. Again, not what we expected when we started doing the study, because Klebsiella is more commonly known as a hospital acquired infection. I've forgotten the second question.
Drug of choice for treating carbapenem.
Yeah. So it's very interesting because, to get antimicrobial susceptibility results from the lab, it can take up to 72 hours. And a lot of these labs are in resource poor settings. So we'll do backtech, then we'll do identification in parallel, we'll do AST. So it takes a while to get that result. So the child gets admitted, they immediately get started with antibiotics. And it's like a cocktail of everything that the physician or the treating physician can kind of give that baby. So we don't use colistin very often because these are very, very little babies. But if we look at the medical charts, we see a lot of use of amikacin. Yeah, and carbapenem is also used. It's just that it's changed after the ASC results get to the doctor. But usually we'll do see amikacin, vancomycin, a cocktail of everything, and meropenems that gets changed later.
Sure. Okay, I just have a couple of questions if you don't mind.
Yeah.
I just think you have like such a rich, first of all, an amazing biobank. We can come back to that, but a rich amount of data and the ability that you have to be able to link the specimen to all of the administrative data. Have you looked at kind of like the geographical spread, maybe not even geographical, but even by hospital spread of the various ST types?
Yes, so that's analysis that we actually just finished. And again, I'm like, oh, I should have added that slide too. So another thing that we were hypothesizing when we were, doing all the genomic studies, looking at key types and STs, first we thought they would be hospital acquired, and we thought even if they're not hospital acquired, maybe different regions of the country will have different sequence types. But we were completely wrong. When certain sequence types take over, they just take over the entire country. So, when we saw SC11 in 2020 and 2021, they were coming from Chittagong, Mirzapur, Sylhet. So these are like different places of Bangladesh that are very far away from each other. I mean, small country, but still far away. And they were all coming with ST11. So it looks like there are these local problem clones that just take over the entire country. And again, we don't understand how they are spreading so quickly and taking over the population of other bacteria. So no, we do not see any specific geographical or hospital-specific clustering of these sequence types.
Do you see any specific clones with respect, and I'm sorry if you mentioned this and I just missed it, with respect to the mortality or like severity of infection?
No, we don't. Year to year, if you look at do just year specific analysis, mortality is already so high, like about 50%. So really, we don't see a very big ST or a clone association with specific clones of mortality. However, what we do see is that, the recent clones that are circulating in the country are causing higher mortality than clones that were circulating 10 years ago, and they could be sensitive or resistant. And there could be a lot of discussions about that, right? Is it the treatment strategy? Are we just assuming they will be carbapenem resistant too, and we're giving them, drugs that might not be appropriate? Are the babies now, because we're also able to save tinier and tinier babies, right? So premature babies are more likely to survive the first couple of days, and they're more likely to get infections and succumb to the infection because they are more vulnerable than others. So the first answer is no. We don't see any clone-specific association with mortality, but we do see an increase in mortality, whether resistant or sensitive strains.
Just going back to, I guess, I don't know if you can answer this question, but because there's limited nosocomial transmission, do you know if there's any screening at all done on the mums for colonization, or is it that like the babies are coming into hospital like already sick, right? I think that's what you mentioned earlier.
Yeah. So no screening is done for Klebsiella on the moms. And also because it's such a ubiquitous bacteria, most likely we'll find some Klebsiella. But what would be really important to answer this question is, swabbing the mom, getting the isolate, sequencing, and then following the baby and seeing what infection the baby gets by sequencing also. So genomics will help us answer that question, but currently no data.
Okay, there's only a couple minutes left, and I just wanted to highlight like the amazing work, considering like the infrastructure that's needed, like the expertise, like that's needed to do this, and just going back to your title. So, like even within Ontario and Canada, there's low resource settings, and there's going to be different challenges. But I think some of the learnings of implementing like a whole genome sequencing program in a setting like yours could be applied to low resource settings in Ontario and Canada. Are you able to share like some of your key learnings, especially like, let me tell you, like we try even at PHO, which is, I would say, a high resourced organization, it's very difficult for us to establish like a very robust biobank. And I think that, part of it's because we have so many isolates, right? But your ability to have a biobank, but then tied to all of this administrative data is incredible. So are there any key learnings from your experience that you could share with us today?
Thanks, Vanessa, for that question. And of course, the credit goes to the founding team who made, Child Health Research Foundation and thought of this idea of biobank. And at a time when, forget genomic sequencing, there's no PCR, right? There's no PFGE. There's no restriction enzymes used regularly, for example. Uh, I think I think one of the biggest reasons that this model has been successful in Bangladesh is because there was a lot of ownership of the hospitals. Because oftentimes we work in silos where people working in the lab works in the lab, people treating patients just treat patients, and we don't crosstalk a lot. I don't know how much of this model we can even expand in Bangladesh. The reason this works, and we discussed this so much because we're like, if we wanted to create the model again in Bangladesh, how would we do it? It has to be from the hospital where it's not only the microbiologist sitting there who's interested in the pathogen, but there are a few champion clinicians who understand the need for it and understand the importance of collecting the required data. And again, a lot of administrative help, like getting the ethical approval to collect those samples, biobank those samples for so many years, and keeping the data is also, very challenging. So we also have, because the hospital is so supportive, the ethical board of the hospital has always supported um, work like this, and we continue engaging with them constantly, and we share all our results. It's not only about authorship, it's about, oh, I think we see an outbreak. So Klebsiella, we are not being able to detect outbreaks, but there are other cases. We detect outbreak, we find results, we immediately communicate with them. There's a lot of crosstalk between hospitals and the labs, and I think that's one of the key successes. And they were never like study specific, like we got a grant and we did it 2 years, the grant is gone and we stop. There's always a sustainability plan so the clinicians don't feel like we're only doing this for research. This is a program that's long term.
Thank you for sharing that. And yeah, wholeheartedly agree. Like you need buy-in, you need champions to get the work done. And I think that's a great way to end today's session. Thank you again so much. It was such a exciting presentation and so wonderful to see what you could achieve in Bangladesh. So as we conclude today's rounds, I would like to extend my most sincere thanks to you, Senjuti, for delivering an amazing, highly informative, and engaging presentation. And of course, a sincere thanks to all of you who joined us today. Your time and active participation are really, really appreciated. You'll receive a brief anonymous survey following today's session. We really encourage you to take a few minutes just to complete it. Your feedback is truly invaluable in helping us strengthen and shape future programming. Thank you once again, and we wish you a wonderful day.